today's class, we are going to discuss with the dynamic state of body constituents and the concept of metabolism. Okay. So already we have discussed about the different types of biomolecules and their biological significance. So what is the dynamic state of body constituents? So we know that the living organisms are made up of thousands of organic compounds or biomolecules. Okay. So the living organisms are made up of, living organisms means, so it includes plants, animals, even microorganisms. So they all are made up of, so different types of organic compounds are biomolecules. Okay, so they are made up of what? So they are made up of biomolecules. Each biomolecule has its turnover. So what is the meaning of turnover? So each biomolecule has its turnover. That means, so the biomolecules are being constantly changed into some other biomolecules. And so they are made in the cells also. That means turnover means, so for example, when you consider a biomolecule, so this biomolecule will be converted into another biomolecules. And the first biomolecule is made again. So that means, so the formation of biomolecules and breaking of biomolecules occurs constantly in a cell. So that we will call it as the turnover. Okay. So the next after the turnover is, so as I told that breaking and formation of biomolecules. So the formation and breaking of biomolecules, nothing but metabolism. That means the sum of total of all the reactions. Either it may be breaking or formation. Either it may be what? Breaking or formation. All these reactions occurring in a cell or in the body, we'll call it as metabolism. Okay. So let us see some examples for the metabolism. So one of the examples. Well, removal of carbon dioxide from amino acids. Okay. So removal of carbon dioxide from the amino acids. So changes the amino acid into amine. Okay. So changes the amino acid into what? Amine group. Is it clear? So here, so the one biomolecule is changed into another. Okay. Next example. A base hydrolysis of a glycosidic bond. So where you can find a glycosidic bond? It is found in the carbohydrate. So the base hydrolysis of a glycosidic bond present in a disaccharide. So one of the examples for disaccharide is the lactose. Okay. So the hydrolysis of glycosidic bond found in the lactose yields glucose and galactose. It yields what? One molecule of glucose and one molecule of galactose. Okay. So this is the example for metabolism. Next is, so these biomolecules are changed into one another. So here a series of linked reactions. That means, so conversion of one biomolecule to another biomolecules. So there is a number of linked reactions are there. So that reactions together, we'll call it as metabolic pathway. Okay. So metabolic pathway can be defined as, so the flow of biomolecules or the metabolites are converted into one another through a series of linked reactions called as metabolic pathway. Okay. So the metabolic pathway is defined as, it is a series of linked reactions. A series of linked reactions through which biomolecules or metabolites are converted into one another. Biomolecules are converted into one another, which is called as metabolic pathway. Is it clear? Next. So the flow of metabolites or biomolecules through the metabolic pathway has a definite rate and definite direction, like the automobile traffic. Okay, so the flow of biomolecules or flow of metabolites through the metabolic pathway has definite rate and definite direction. Hence, it is called as dynamic state of body constituents. Is that clear? Okay. So, what is the dynamic state of body constituents? So, it is flow of metabolites. Flow of metabolites. Flow of metabolites through the metabolic pathway. Through the metabolic pathway in a definite rate. In a definite rate and definite direction. Okay, so definite direction is called as dynamic state of body constituents. Dynamic state of body constituents. Is it clear, students? So here what happens? A specific amount, definite rate, a specific amount biomolecules will flow in a particular direction, just like the automobile traffic. So that we'll call it as dynamic state of the body constituents. So all this metabolic pathways are catabolized. So catalyzed by a catalyst. Okay. So all the metabolic pathways are catalyzed. So I'll tell you the meaning of catalyzed. So they are catalyzed by a catalyst. They are catalyzed by a catalyst. Catalyst. What is the meaning of catalyst? So catalyst is nothing but, so it is a chemical, so which accelerate the rate of reaction. Okay. So catalyst is nothing but which accelerates. Accelerates means which increases the rate of reaction is called as the catalyst. So one of such catalyst is the enzymes. Okay, enzymes. So all the metabolic pathways are catalyzed by catalyst. A catalyst is nothing but, so which accelerate the rate of reaction like enzymes. Okay, so and enzymes are called as biocatalyst. Okay, so enzymes are called as what? Biocatalyst. Why they are called as biocatalyst? So these enzymes are, so they act as a catalyst, or they possess catalytic power, as well as all the enzymes are made up of proteins. Okay, what? So all the enzymes are made up of what? Proteins, and they possess catalytic power. Catalytic power is called as the biocatalyst. Is it clear? So all the reactions which is occurring in the living organisms, so it is catalyzed by the enzymes. Okay, so even the simple reaction like, so where the carbon dioxide dissolving in the water, so in case of living systems, is also catalyzed by a enzyme. So let us see that example. Okay, so all the reactions which are occurring in the living organisms are catalyzed by the enzymes. So one of the example is, whereas the simple reaction like the carbon dioxide dissolving in water. So the formula of carbon dioxide is, it is CO2 and water is H2O. So dissolving in water, that is carbon dioxide dissolves in water to form carbonic acid. The formula is H2CO3. So this is carbonic acid. So even such a simple reaction is also catalyzed by the enzyme called as carbonic anhydrase. Okay, so the enzyme called as what? Carbonic anhydrase. Okay, so that means all the reactions which are occurring in the living organisms are catalyzed by the enzymes. Okay, so this is regarding the dynamic state of body constituents. So let us see the metabolic pathways. As I told that metabolic pathways where a series of reactions are involved. So there are two types of metabolic pathways in case of living organisms. So let us see that. Okay. So the metabolic pathways are of two types. Okay. So one is called as catabolism, another one is called as anabolism. So the types of metabolic pathways are of two types. So the first one is, let us take up with the anabolic pathway. So these anabolic pathways are also called by another name, that is nothing but biosynthetic pathways. Okay. So it is also called as what? Biosynthetic pathway. Okay. So another pathway is the catabolic pathway. Okay. The second type is catabolic pathway. Catabolic pathways. Okay. The second one, catabolic pathway. Okay. So first let us discuss with the anabolic pathway. So anabolic pathway is nothing but where the formation of complex biomolecules takes place. Okay. So the formation of complex structures takes place. Such type of metabolic pathway is called as anabolic pathways. Okay. So I can define anabolic pathway as it is a type of pathways in which the simple structures combine to form a complex structure is called as anabolic pathway. Okay. So what is anabolic pathway? So it is a type of metabolic pathway in which simple structures combine to form complex structure is called as biosynthetic pathway or anabolic pathway. So the formation of biomolecules here, it consumes energy. So hence anabolic pathway, so it is a type of metabolic pathway where energy is consumed. Okay. So energy is consumed. So let us see the example for anabolic pathway. So with the formation, isn't it? So here formation of cholesterol takes place in the liver. So here the precursor is acetic acid. Okay. The simple molecules like acetic acid is converted into a complex structures like cholesterol. So this process, where does it take place? It is occurring in the liver. Okay. Or even you can take the formation of glycogen also. Okay. So this is the example for anabolic pathway. Next, catabolic pathway. Catabolic pathway means breakdown. So it is a type of metabolic pathway where simple structures are formed due to the breakdown of complex structures. Okay. So catabolic pathway, you can define it as it is a type of metabolic pathway. It is a type of metabolic pathway where simple structures are formed due to the breakdown of complex structures. Due to the breakdown of what? Complex structures. So here, when these complex structures break down into simple structures, it yields or it produces energy. So it produces energy. Or in simple terms, we can tell that. So catabolic pathway where the degradation of complex structure into simple structure which yields energy. So the energy produced during degradation is trapped by the living organisms in the chemical bonds of ATP. Okay, so the energy produced during the degradation is trapped by the living organisms in the form of ATP. So we know that ATP stands for adenosine triphosphate. ATP is nothing but what? Adenosine triphosphate. Adenosine triphosphate. So when there is a requirement of energy or when there is a need of energy, so the chemical bonds of ATP breaks down and the energy is released. Okay. So when there is a need, what happens during the need? So the ATP breaks down. ATP breaks down and releases the energy. So which is utilized for performing the osmotic functions of osmotic work or mechanical works. So which can be utilized for performing what? Osmosis or any mechanical work. Any mechanical work. So this is how the energy released during the catabolic pathway is used for the different types of functions or work. Okay. So let us know about some more about the ATP. So as I told that ATP is what? Adenosine triphosphate. It is also called as universal energy carriers. It is called as what? ATP is called as universal energy carriers. Universal energy carriers. While discussing about RNA, I told you. So whereas higher nucleotides can function as a energy carriers. Okay, that is, so which has more than one phosphate group, we consider them as a higher nucleotide. Whereas ATP consists of 3 phosphate groups. So hence it is one of the higher nucleotide which acts as a energy carrier. So it is also called by another name, that is energy currency of a cell. So another name for ATP is, it is called as what? Energy currency. Energy currency of a cell. Okay. So adenosine triphosphate is a non-genetic nucleotide. What it is? It is a, ATP is a non-genetic nucleotide. Non-genetic nucleotide. So what and all ATP is made up of? As it is a nucleotide, it is made up of one pentose sugar. The pentose sugar name is ribose. So the pentose sugar name here is the ribose. And so it is made, it consists of adenine, the nitrogenous base, adenine. Along with that, here the 3 phosphate groups are found. So along with that, it consists of 3 phosphate groups. So ATP is a non-genetic nucleotide. It is made up of ribose, adenine, and a row of 3 phosphate groups. Okay. And so this ATP is preferably formed during the cellular respiration. Okay. So it is formed during where? So it is formed during the cellular respiration. Cellular respiration. So now let us see that the living state of biomolecules. Okay. A living state and metabolism. So in the living state and metabolism. So here, a living state. So in living organisms, okay. So in living organisms, living state we are discussing related to the metabolism and biomolecules. So in living organisms, there are tens and thousands of biomolecules are present in a various concentration. Okay, so living organism consists of different types of biomolecules, and these different types of biomolecules are present in a various concentration. They are present in the various concentration. Let us see the example. So for example, for that is, let me take the first example as a glucose. So the glucose is, it is 4.5 to 5 mm, that is millimolar per deciliter in a healthy individual. Okay, so the glucose is how much? It is 4.5 to 5 millimolar per deciliter in a living organism. So that is in a healthy individual. Okay. So the next is, let us take the example of a hormones. Let's take the example of a hormone. So whereas hormones are measured in nanograms. Okay. So hormones are measured in nanograms per ml. Okay. So here you can see that. So each biomolecules are ranging in a various concentrations. Is it clear? Okay. The next is, so the living organisms. So living organisms always they are present in a steady state. Okay. So living organisms they are always present in a steady state. So this steady state is a non-equilibrium state. Steady state is a non-equilibrium state. That means, so when the living organisms are in a steady state, so it is a non-equilibrium. That means, so when the organism is living, some or the other reactions, so metabolism will be going on in a cell or in an organism. Hence, so it is a non-equilibrium state. Okay. So whichever reaction, either it is a physical or chemical reaction, always a reaction will tend to move towards the equilibrium. But so in case of living organism, if it reaches equilibrium means the cell is not performing any function. That means it is not living. Okay. That means living state is a steady state, but so it is a non-equilibrium. So it is a non-equilibrium condition. Okay. And so they are under metabolic flux. What is the meaning of metabolic flux? The rate of turnover. Okay. So rate of turnover, we'll call it as metabolic flux. What is the meaning of rate of turnover of biomolecules? That is, so the amount of formation and breakdown of biomolecules occurs is called as rate of turnover. Nothing but metabolic flux. So a living organism means always it will be in a steady state. A steady state is a non-equilibrium condition. So this is regarding the dynamic state of the body constituents. So let us see the introduction of enzymes. So next topic is the enzymes. Just now we studied some points related to the enzymes. So as you know that it is a biocatalyst and it is made up of what? Proteins, and it increases the rate of reaction. Isn't it? So enzyme is nothing but what? So the major function is it acts as a catalyst which increases the rate of reaction. Isn't it? So let us study that. So enzymes. So enzymes can be defined as, so these are the biocatalyst or biological catalyst, so which increases the rate of reaction or which accelerates the rate of reaction without undergoing any changes. It's called as what? Enzymes. Okay, enzymes can be defined as what? So it is the biological catalyst. Enzymes are biological catalyst which accelerates rate of reaction. Rate of reactions in a speedy manner. In a speedy or in an efficient manner without undergoing any changes. Without undergoing any changes is called as the enzyme. So the enzymes were discovered by Edward Buchner and J.B. Sumner. Okay, so enzymes were discovered. So they were discovered by Edward Buchner and J.B. Sumner. Okay, so these are the two scientists who discovered the enzymes. And the term enzyme has been derived or coined by, so the term enzyme has been derived or coined by Kuhne in 1878. So the meaning of enzymes means in yeast. Okay, so meaning of enzymes is in yeast. So meaning of enzymes is in yeast as they were discovered in the yeast. Okay. So this is regarding the introduction of enzymes. So when you see enzymes, they are chemically made up of carbon, hydrogen, oxygen, nitrogen and sulfur. Okay. And all the enzymes are proteins. This is important. All the enzymes are proteins. But all the proteins are not enzymes. All the proteins are not enzymes. It's an important thing. So we know that. So all the enzymes are made up of what? Proteins. But all proteins are not enzymes, because a protein can form into an enzyme, or it can form into any structures like hair, nails, like that. So or it can form into hormone. Okay, etc. So due to which, so all the enzymes are made up of proteins, but all the proteins are not enzymes. Okay. So in some cases, the enzymes are not only made up of proteins. So in some cases, some nucleic acid act as an enzyme. What happens? So the nucleic acid act as a enzyme. So that we'll call it as ribozymes. That we'll call it as what? Ribozymes. Okay. So important thing. Ribozyme means nucleic acid acting as a enzyme. Is it clear? Okay. Next. So when you consider a functional or an active enzyme, it is made up of protein part. Okay. A functional enzyme is made up of a protein part called as apoenzyme. Called as what? Apoenzyme. And so it is also made up of a non-protein part. Non-protein part called as coenzyme or cofactor. Coenzyme or cofactor. Okay. Together, that is protein and non-protein part together produces the functional enzyme. Produces what? A functional enzyme. Okay. So here the protein part is apoenzyme. Non-protein part is cofactor. Together produces a functional part is called as holoenzyme. Holoenzyme. Next. So these enzymes function as a catalyst. We know that, which increases the rate of reaction. That is totally due to the presence of active site. So these active sites are nothing but crevices. That is cracks or pockets on the surface of enzyme. See, for example, if you consider this as the enzyme, so a small pocket-like structure is called as active site. Is called as active site. So, to which a substrate binds. To the active site, a substrate binds. Later, the substrate will be converted into product. That means, so here, so active site is nothing but what? The pocket on the surface of the enzyme is called as active site. So, to which the substrate binds and which will get converted into product. So, the catalytic activity of the enzyme is controlled by what? So, the active sites. Is it clear? So, this is regarding the active site. So, in some cases, so these proteins are actually they are sensitive to temperature. But whereas thermophilous organisms, thermophilous, what is the meaning of thermophilous? Thermophilous means the organisms which grows in a extensive temperature. The organism which grows in the extensive, that is high temperature, we call it as thermophilous organisms. Okay. So, in case of thermophilous organisms, that is the organism growing in the extreme temperature. Extreme temperature has the enzymes. Okay. So, which are thermostable. Okay. So, actually the enzymes are sensitive to heat or temperature. But whereas in case of thermophilous organisms, so which are growing in the extreme heat or extreme temperatures like 80 to 90 degrees Celsius, So they have a thermostable enzyme. So that enzyme is called as top polymerase. So top polymerase. So this top polymerase has been isolated from a thermophilous organism called as. So Thermophilus aquaticus. Okay. So top polymerase is isolated from the one of the organism called as Thermophilus aquaticus. Is an organism from which so the top polymerase has been isolated. So this is of commercial use. So this is a type of DNA polymerase. So this is used in the PCR technique, that is polymerase chain reaction technique. So in detail we'll study about this in the second year. Okay. So now chemical reactions. Chemical reactions. So the chemical reactions. So the chemicals can undergo a physical change or chemical change. Okay. So the chemical compounds can undergo physical process or chemical process. So for example, when you consider a physical process where in case of physical process, there is no breakdown of bonds. So they will be changed, there is only change in the shape. Okay. So here the change in shape takes place, but there is no breakdown of bonds. There is no breakdown of bonds. So one of the example for physical process you can take is where the ice melts into water. The water turns into water vapor, where you can see the change in the state. Okay, like ice melts into water, or the water changes into water vapor is the some of the examples for the physical process. So the second one is the chemical process where in chemical process there is a breakdown and formation of the chemical bonds takes place. Okay, so here what happens? Breakdown and formation of chemical bonds takes place. Okay. See, one of the example is, I'll take the example of a barium hydroxide, BaOH twice, combines with sulfuric acid, H3SO4, to produce. The product here is, see, the bond present between the barium and hydroxide will be broken, and so the new bond will be created between barium and sulfate, results in the formation of barium sulfate and 2H2O. Okay, so there is a breakdown of bonds takes place and formation of bonds takes place. So this is one of the example for inorganic chemical process. So let us take the example for the organic process, that is where the starch breaks to form glucose. Okay, so as we know that, so starch, glycosidic bonds will be present. So which will be broken to form what? It will be broken to form glucose. It will be broken to form glucose. So here, rate of reaction, rate of metabolites or biomolecules. What is the rate of reaction? So rate of reaction is nothing but, so the number of product formed in a unit time. Okay, so number of product formed in a unit time, or the amount of product formed in a unit time. So it is expressed as delta P divided by delta T. Delta P represents the amount of product and delta T represents the time take. Okay, so the rate of reaction is, it is if we specify in a reaction, if we specify the direction, the rate of reaction, when we specify the direction, it can be called as velocity. Okay, what? So when we specify the direction for rate of reaction, it can be called as the velocity. Okay. And the next is, so in case of here, there is a general thumb rule. That means when we increase the temperature. So as I told that, so enzymes are sensitive to the temperature. So when you increase the temperature to 10 degrees Celsius, so the reaction will be doubled, or so it will be doubled or reduced to half. Okay, so that is the thumb rule followed by the biomolecules or the enzymes. So here one of the example is carbon dioxide combining with the water. The simple reaction which we have taken and results in the formation of what? Carbonic acid. Okay. So let us see this reaction in the presence of enzymes, in the absence of enzyme. So the first one is carbon dioxide combines with the water to form carbonic acid, H2CO3. So here it is in the absence of the enzyme. So how many molecules of carbonic acid can be formed here? It is around only 200 molecules. That is also time taken here is per hour. Okay, so let us see the same reaction in the presence of enzyme called as carbonic anhydrase. So when the enzyme is there, so which is the enzyme? That is carbonic anhydrase. Remember, so this is the one of the fastest enzyme. Among all the enzymes, which is the fastest enzyme? That is carbonic anhydrase. So when this enzyme is there, so the amount of molecules formed are 6 lakh carbonic acid are formed. That is also per second. Okay, so you can see that. So in the presence of enzyme, the rate of reaction is increased 10 million times. Okay, so that is the efficiency of the enzyme. So not only this, even the reaction, the enzymes can be involved in a metabolic pathway. Metabolic pathway means a series of reactions which are linked with one another. So in a metabolic pathway, for example, let me take the example of a glycolysis. So in case of glycolysis, so the glycolysis, so glucose. In case of glycolysis, what happens here? Glucose is turned into or converted into 2 molecules of pyruvic acid. It is converted into 2 molecules of pyruvic acid. Okay, so this conversion actually requires nearly 10 steps. So each step is catalyzed by a enzyme. Okay, so when there is a addition of 1 or 2 steps, there is a addition of 1 or 2 extra steps along with the 10 steps, it produces a new product. For example, so this pyruvic acid, in case of pyruvic acid, in the skeletal muscles, what happens in the normal condition is this pyruvic acid will be converted into acetyl-CoA. Will be converted into what? Acetyl-CoA. Later the acetyl-CoA is converted into carbon dioxide and water. So under aerobic condition, that is presence of oxygen. Okay. The next, so the same pyruvic acid under anaerobic condition. So this is under anaerobic means in the absence of oxygen. What happens here? So the pyruvic acid will be converted into, lactic acid. So due to which the pH of the muscle cells will be increased, which causes the stinging effect. So the same reaction in case of yeast. So under anaerobic condition, pyruvic acid will be converted into alcohol. So this process we'll call it as what? Fermentation. Okay. So this is regarding the glycolysis metabolic pathway. Okay. So let us see some of the questions related to the topic. So the first question is, enzymes are proteins, hormones, vitamins, none of these. We know that enzymes they are all made up of proteins. The answer is A. Second one, an organic substrate bound to the enzyme and essential for its activity is called holoenzyme, apoenzyme, isoenzyme, coenzyme. Okay, so the non-protein part bound to the protein we'll call it as what? Coenzyme. So the answer is D. Next is, the term enzyme was coined by Edward Buchner, Sumner, Kuhne, Mueller. So the term was coined by Kuhne, but it was discovered by Edward and Buchner. So here they have asked the term, it is Kuhne. The answer is C. Fourth one, the fastest enzyme in the world is carbonic anhydrase, pepsin, lysozyme, amylase. The answer is carbonic anhydrase. So this is regarding today's class today. Thank you.